So these are the results of the UV treatment experiment. Probably most importantly right now, we can see the results of our dilution series. So I've arranged them from left to right. This is the undiluted, 10 to the minus 2, 10 to the minus 4, 10 to the minus 6, 10 to the minus 8. And the plan was for each of these plates, as we go from left to right, to have 100 times fewer cells than the one before it. So we can get in here to count these in a little bit. There's a little bit of spreading artifact here. You can see some patches that are empty and some patches that are really dense. Comparing these are supposed to be the same dilution. The bottom one appears to have fewer cells, whereas these ones, it looks like they're about the same number of cells. So not quite sure what's going on there. Really, when we're looking at number of cells, this density of cells is just unreliable. So the cells are growing too closely together. They've grown up into colonies. So I guess sometimes I interchange cells and colonies. I should be talking about colonies here. So a colony is a more or less a perfectly circular blob of cells on here, and that originated from a single cell. So one cell was put onto that petri dish and grown up, and as it grew, it just divided and filled up that space in every direction from the starting cell. So one cell becomes one colony, and these are important genetically because each of those would be genetically, potentially genetically unique. So if we are generating mutants, then each colony could be its own mutant. Could be that in a lot of cases we're not going to see anything that is evidence of a mutant, but important to look for them. That's what we're going to do. So I want to point out also, this was our target dilution, 10 to the minus 4, and I had multiple replicates of 10 to the minus 4 that went into the UV treatment. So we can count these. These are definitely a countable number of colonies. Maybe around 100. I'll count these and let you know what I find out. These colonies, I think, are just under where I would say they are reliable, or maybe just over, I should say. So these colonies are just so dense that they bump into each other, and I can't quite tell where one stops and the next one begins. So that's really one of the big reasons we do the dilution series, is to separate those out so that we can count them individually, so we can treat them individually, and we're going to grab them and expose them to some treatments moving forward. Didn't really spend much time talking about these. These are the next dilution up. So if I was to guess, I'd say there's about 100 cells on these 10 to the minus 4 plates. And if we move over to the 10 to the minus 6, there should be 100 times fewer cells, which is basically, there's one. One colony has grown here, and no colonies have grown here. Basically just down to the chance that one cell made it through all that dilution and got plated on here. So it's probably fewer cells than we would usually like at the 10 to the minus 6 dilution, but we can work with what we have. And then of course 100 times more dilute than that one, we really wouldn't expect to see anything. Although it's always possible that one cell squeaks through all those dilutions and makes it onto the plate. So kind of as predicted, 10 to the minus 4 is our most reliable dilution. It's got a countable number of cells. And with these 5 replicates of the UV treated plates, we have enough that we can explore what's going on with those colonies.